Welcome. This is the Chemistry, Biology and Math Revision Hub. Today we'll be doing the Pearson Edexcel International A Biology, Unit 4 for May 2021. The paper is 90 marks, so I will begin off with question 1. Question 1 says, mosses are species of plants that have genomes that enable them to survive in their habitats. A group of students investigated the mean number of plant species in 4 habitats. The habitats that they investigated were number 1, of course, fields that were cut regularly, and then the next part, fields that were not cut, the middle of woods, and the edge of woods. The graph shows some of the results from this investigation. So as we can see from the key, we can see there are 4 different types of plants that were investigated. So on the vertical axis, we have the mean number of plant species, and the horizontal axis, we have the habitats. So here they've given us 2 habitats, the one that was regularly cut and the one that was not. And we can see we have more species growing in the area that was not cut regularly, while here we have only 2 species in the area where there was regular cutting. So we have fewer species. However, we have a higher number or a higher mean number of plant species in the field that was cut regularly. So when you look at this graph, you can see this species should be, in this case it's the grass, it should be fast growing because it's in an area that is regularly cut, but the mean number is really much or bigger. It means it's fast growing, and because it grows faster, maybe the others like the large bushes as well as the small bushes that are not present in this field that is cut regularly, it's because they are slow growing. So those that are fast growing are going to grow faster before the next cut, and then in comparison to those that grow slowly. When we look at these ones, the area that is not cut, they are many species. However, the mean number is not as much. That could be due to competition existing between the organism or between the species of plants. So I will go to the first question regarding this graph. It says, which row of the table shows the type of factor that affected the plant species in 2 of these habitats? So the area cutting of the field regularly is of course biotic, and then competition with trees for water, that is also biotic. So here it should be D, the answer for the first question. Then the next part says, what is the percentage increase in the number of species of moss between the fields cut regularly and the fields not cut? Allow me to go to the previous page. Remember, we are looking at the number of mosses in between the area that is regularly cut and the area that is not. So as you can see here, mosses are the ones in black. So I am looking for that and that. Here you can see for that one, the number is 1. And initially for this one is about 0.3. So it moved from 0.3 to 1. I will go back to the previous page to try and calculate the percentage increase. So the percentage increase should be the new number minus original number divided by the original times 100. So it will be 1 minus 0.3 divided by 0.3 times 100, which gives us a percentage of 233.3%, which gives us C as the answer. The next part says, suggest reasons for the differences in the mean number of plant species in fields cut regularly and fields not cut regularly. So the key thing is, in the field that was cut regularly, of course, it allowed the fast-growing grasses to grow rapidly, using up, of course, there was no, if you're cutting the field regularly, there is not going to be competition for light because light is going to be available since all the plants in that area are going to be cut and they're going to be at the same level. There will be enough light available to all of them in order to grow. There will be enough, of course, minerals in the soil because the ones that are cut are going to decompose and they will provide minerals and nutrients for the next phase of growth. So I say the cut fields have more grass than mosses. So this is a fact from the graph. This is because grass grows faster. Yep, grass grows faster. So when it grows faster, it's going to reduce the light and minerals available for other plants because as it grows faster, it's going to use them. And the increase in height or increase in size is going to cover the others not to experience the same light intensity as the grass experienced before, meaning there is going to be kind of competition for the other resources between the other plants as well as the grass. So the grass actually outcompetes them for these resources. Then the next part, the bushes with less light, of course, you remember I said grasses grow faster, but the bushes are actually growing slower. It's because but they are slow growing, and by the time they grow, most of they are out competed, or the nutrients are already taken out, so their growth will not be as fast as the others. And another thing, since they grow slowly, by the time they are trying to grow, then the next cutting phase occurs, and then they have to grow again. So it's kind of a disturbance for them. That is why the bushes, when they have less light, they will grow slowly, and they will not have enough time to grow before the next cutting phase. So they are not given the opportunity to increase in number. So I'll go to the next question. The next question says, in another investigation, the genome size of 30 species, so these are 30 species of moss, was determined. Each species of moss belonged to one of two classes of moss. So they say the graph shows the frequency of each genome size in the class of moss, and in some class of moss 1, basically class 1, and there is another class 2. So they say class 1. So here we have the class 1 mosses and we have the class 2 mosses. As you can see, I already wrote them in here. Here we have the frequencies 1 because of that. Here frequencies 4, frequencies 9, frequency 7, and I have put them here 1, 4, and 2. So the first question says, which does P represent? If you guys see, P has the highest frequency, and as we know, the number with the highest frequency is usually the model number. So in this case, it should be the mod genome size of this histogram. That is why D is the answer. It's the mod genome size of the histogram. This is a histogram. It is not a bar chart, because a bar chart and the histogram have different. They have different labels on the axis, basically. So I will continue to the next part. One genome size is missing from the graph for class 2 mosses. Calculate the frequency of the genome. So they're telling us that there is a genome class missing here. However, we need to find the frequency. To find the frequency, since we were told that there were 30 species of moss, and you can see in this area we have a 9 plus a 7 plus a 4 plus a 1, which gives us 21. And the 2 plus a 4 plus 1, which gives us 7. 21 plus 7 gives us 28. And the original total 30 minus 28 gives us a 2. So this should be a 2. The missing class should have genome size or genome frequency equal to 2. I will go to the next part. The next part says, the mean value for the genome size of class 1 mosses was 0.449 AU. And the mean value for class 2 mosses was 0.920 AU. They say calculate the ratio of genome size of class 1 mosses to the genome size of class 2 mosses. So here what we have to do, it's a ratio basically. So all you need to do is list 0.449 to 0.920, and then divide the bigger one by the smallest, so that you find a ratio of 1 to something. And since this is 1 to 2.049, we cannot write it as decimal because it has to be a ratio. So the answer should be 1 to 2, which is that. Lastly, here we have, it was suggested that the chromosomes in class 2 mosses were found in pairs. Give the evidence that supports this suggestion. So if you want to know that the chromosomes were found in pair, when you look at class 2 mosses, they have almost twice the genome size of class 1 mosses, and that is the reason as to why they are found in pairs. So this is the confirmation that they are found in pairs, is because class 2 mosses have almost twice the genome size of class 1. When you look at this, of course, 2 to 1, so it means the others are in pairs and this one may not be paired. So that is one reason you could give for that. This brings us to the end of question 1. Let us move on to question 2. Question 2, chloroplasts and mitochondria are two organelles found in some plant cells. They say compare and contrast the structures or the structure of a chloroplast with the structure of a mitochondrion. So here, when they ask you to compare and contrast, it means they want you to give the similarities as well as the differences. So I began off with similarities. As we know, both the chloroplast and the mitochondrion have circular DNA. They both have double membranes, the outer membrane as well as the inner membrane. And they both have ribosomes. Ribosomes for protein synthesis. They have the circular DNA as well as the double membranes. So those were the similarities. And the differences are, inside the chloroplast we have we would say the inner space, which is called the stroma, while in the mitochondria we have the inner space called the matrix. For the chloroplasts, they have thylakoids. Of course, these are the sites of photosynthesis. But in the mitochondria, we have a folded inner membrane called the cristae, the site for the oxidative. Of course, respiration involving aerobic respiration, basically. The chloroplasts have starch granules, while the mitochondria do not. So if you compare, there is an image I inserted in here. You can see this is the cristae in the mitochondria. That is, of course, we have the matrix, and then we have the stroma in the chloroplast. They have DNA, as you can see for both of them. There are ribosomes here, there are ribosomes in here, and then we have the two membrane. You can see the outer membrane, and then the intermembrane space, and the inner membrane. Here also outer membrane and the inner membrane. So you can see the differences as well as the similarities between the chloroplast and the mitochondrion. So I will move on to the next part. So part B says the light-dependent reactions of photosynthesis can result in either cyclic or non-cyclic photophosphorylation. They say the diagram shows cyclic photophosphorylation. They say complete the diagram by writing the correct word or words on the dotted lines labeled D, E, F, G, H. So one thing we need to know about cyclic photophosphorylation is that it's going to occur in photosystem 1, or what we call PS1. So when you're in PS1 here, an electron is going to move. It's going to be excited, of course, to a higher energy level. And then as it transitions, the electron transitions, is going to lose energy as it transitions through the electron carrier. So you can see here E is the electron carrier. You can see they could be proteins, or you can call them cytochromes, where the electron is going to be carried to lower levels as it loses energy. Now, this electron is going to be, the energy lost is going to be used to convert ADP into ATP when ADP and PI are combined to produce ATP. So when we go down here, the next question says, name one molecule that is produced in non-cyclic photophosphorylation and non-produced in cyclic photophosphorylation. Of course, this is going to be reduced NADP. Both cyclic and non-cyclic produce ATP. However, reduced NADP is produced in non-cyclic photophosphorylation. So that is the answer. I continue to the next part. They say describe the role of photolysis in non-cyclic photophosphorylation. Photolysis is when a water molecule is split, releasing hydrogen ions as well as electrons. So this is done by the light that comes from the sun, basically. So remember, they're asking us the role of photolysis. So you have to talk about how it occurs, and when it occurs, the products that come out of it, where are they used? So I said photolysis splits the water molecule, releasing 2 electrons and 2 hydrogen ions. The electrons replace the excited electron from photosystem 2. I hope you guys remember we have photosystem 2, we have photosystem 1. This electron will go and then come down here, and those will go, and then maybe they could be, this is PS2 and this is PS1. And as you have seen in PS1, they were recycling photophosphorylation, so the electrons could be recycled and recycled here, but in that process, when the electron leaves here for the first time, we need something to replace the lost electron. So that electron is going to come from the photolysis of water, as water molecule is split, replacing the electron taken away from photosystem 2. The next part, I say the hydrogen ions reduce NADP to produce NADPH. Of course, this is correct. A proton is going to be taken up by the NADP, and then NADP will become reduced so that it can be taken later on into the Calvin cycle. This brings us to the end of question 2. Let's continue on to question 3. Question 3. Lions, giraffes, and acacia trees are found in the African plains. The diagram shows the energy in the trophic levels that these organisms occupy. So we see the lion at the top, then we see the giraffes, and then there are trees which are the acacia here. The first question says, give the meaning of each of the following terms using the information in the diagram to illustrate your answer. The first one is a habitat. So they ask, what is a habitat? Of course, a habitat is a place where organisms live, or where an organism lives. However, this question also asks you to use they say use information from the diagram to illustrate your answer. So my illustration or example was the African plains. The African plains is a place where a lion, giraffe, and the trees are found. So we can say the African plains. This is a habitat for these organisms in the diagram, basically. So you have to give some form of example or reference to the diagram that is given to you. Now the next part is a population. A population is a group of organisms of a same species living in a particular area. So again, let me come back to this. I can say a population of giraffes. So the population of giraffes is 44,988 and so on. Here I say that population of lions in the African plains. They are the same species. They are living in the same area. They are a population. Let me go to the next page. A community. A community is a group of organisms of different species interacting in a particular area. So in this case, the community involves the lions, giraffes, and the acacia. They have to be different species, as well as the acacia trees in Africa, of the African plains, of course. And then lastly, they asked us about a niche. The role an organism plays in a habitat or in an ecosystem or in an area, that is what a niche is. So in this case, the acacia trees provide shade for the lions, or they can provide food for the giraffes, and then lions keep the giraffe numbers in check, or they control the giraffe numbers. So lions are predators, that is their job. Acacia trees provide a habitat, and then giraffes are basically herbivores. They feed on the trees, on the acacia trees as well. The next question says, calculate the efficiency of energy transfer between trophic level 1 and trophic level 2. So if you compare, I want to take you back again, trophic level 1 and trophic level 2. Trophic level 1 is that. This one here, 5, that. And trophic level 2 is that. So if we want to know the energy efficiency, we have to get the energy transferred. Let me take you back down here. The energy transferred divide by the energy that was available, which is that times 100, and this gives us 8.42, that's the percentage we were looking at. So this brings us to the end of question 3. I will move on to the next question, question 4. Question 4. Madagascar is a large island of the southeastern coast of mainland Africa. Lemurs are endemic to Madagascar. There are many different species of lemur, all of which evolved from one common ancestor. The common ancestor is thought to be a primate that was carried across the sea from mainland Africa on a raft of vegetation. The photograph shows 2 different species of lemur, Sifaka and Indri, found in the same region of Madagascar. So we see this is Sifaka and that is Indri. The diet of Sifaka is mostly seeds, but also includes fruits, flowers, and some types of leaves. The diet of Indri is mainly leaves. So Sifaka feeds on seeds as well as leaves. Okay. They say explain the types of speciation that have taken place in the evolution of Sifakas and Indri in this region of Madagascar. Now, when we refer back to the introduction of the question, they said their ancestor is presumed to have been a primate that was brought on maybe a vegetation raft onto the Madagascar area. So it means since they came from the same parent or same ancestor, it means there was allopatric speciation that occurred. Maybe that ancestor was part of another group, so that ancestor separated from the original group. So that separation was allopatric, meaning there is geographical isolation from mainland Africa to Madagascar. So they were separated from the original, maybe siblings or original group. So I say because the ancestors of both Sifaka and Indri came from mainland Africa, allopatric speciation occurred due to isolation from of their ancestor from mainland Africa. So here you get a mark for talking about allopatric speciation occurring initially. And again, remember they say types of speciation, types. It means it's not going to be only sympatric, it should be sympatric as well as allopatric. So now when we continue, after their ancestor arrived in Madagascar, over years we see there are new species that have arrived or that have been formed from that ancestor. So it means sympatric speciation has occurred within this area. So I said with Indre and Sifaka to be categorized as different species in the same area, sympatric speciation must have occurred, and this created different species which have evolved to feed on different diets. So they have evolved to feed on different diets, maybe because of their lifestyle or because of their preference, or within the same area. Since there has occurred some sympatric speciation, their behavior could have changed, leading them to prefer different types of meals, and that kind of makes them seem different species. Basically, they are different species because they have evolved within the same area. So we can say sympatric speciation has occurred. I will go to the next page. This part says, lemurs have been found in another island called close to Madagascar, and scientists have used DNA profiling to show that these lemurs originated from those on Madagascar. Explain the role of polymerase chain reaction, PCR, in DNA profiling. Polymerase chain reaction amplifies DNA. So what we need to know is if we get smaller quantities of DNA, or if they are collected, PCR amplifies it and helps us to generate larger quantities of DNA that can be used for research, basically. So I say, if small quantities of DNA are collected, PCR enables amplification of DNA, which creates more copies of DNA, and this can be used in gel electrophoresis in order to carry out DNA analysis or to successfully analyze DNA. So that is very important. PCR amplifies DNA, creating larger volumes of DNA, which can be used for a variety of research in order for scientists basically to be able to carry out their role or their job. The next part says, explain how DNA profiling could show that lemurs originated from the lemurs on Madagascar. When we use gel electrophoresis, and again remember PCR just amplifies, it cannot help us to basically tell us the fact that we're looking for. So what can help us is when we do gel electrophoresis and compare the patterns of the bands produced. So I said use gel electrophoresis. It can be run on the two groups. And again, we have two groups, those still in Madagascar and those in the neighboring Madagascar. We will get their DNA samples. We'll use gel electrophoresis. We will run and then look at the patterns produced. If there are similar bands that are produced, then we can be able to make a conclusion. So here, if the lemurs were genetically similar, it means they will have more patterns that are similar. But if they were not, it means that the two groups would have originated differently, because if the patterns are not similar, it means then we can make a conclusion that they are not the same species. They did not originate from the lemurs of Madagascar. Maybe they would have originated from somewhere else. The key thing here is to know that you can use gel electrophoresis, look at the bands produced for both the lemurs you're trying to analyze, and then look at the similarities, meaning the bands that are similar, and look at the differences, meaning the bands present in the DNA sample of one but missing the DNA sample of the other. If you see patterns of less similarity and more differences, then it means they did not originate from the same ancestor. However, if you see many patterns or many bands that are similar or present in both, then it means there is greater similarity, and therefore they could have originated from the same ancestor. So this brings us to the end of question 4. I will move on to question 5. The time of death of a person can be estimated in a number of ways. The time of death can be estimated using the length of insect larvae. What is the name of the method that uses insect larvae to establish the time of death? This method is called forensic entomology. As you can see, dendrochronology is not, because it's observing the tree rings and trying to date basically the age of trees and so on. The other species diversity is talking about the number of different species. You measure the numbers of different species in an area, and epigenetics is about changes, studies on changes in gene expression. So the answer should be C, forensic entomology. Part B says, the larvae of one species of blowfly can be used to estimate the time of death. The graph shows the mean length of larvae from this species incubated at 10.62 degrees Celsius. So here we can see, observing the graph, we see on the y-axis we have the mean length of larvae in millimeters, and on the x-axis we have the time after the larvae has hatched from the eggs in hours. So observing here, we can see that from this to here, actually we have error bars all over. All the results have error bars, and that shows us that the results were repeated. So the experiment was repeated, a mean was calculated, standard deviation was calculated, and then the results were plotted, which is actually good. It shows us that the experiment can be reliable because there were repeats carried out. However, from the beginning until about this, we can see the error bars are really small, meaning the deviation between the highest and the lowest is not that great, meaning the results can be reliable. However, after this to the end, we can see the error bars, or the gap between the maximum and the minimum, is actually big, meaning there is greater deviation, and therefore the results may not be that reliable as, of course, as the length of the egg, as time passes by from the time which the eggs were actually hatched. So results may not be dependable after 45 hours. So the first question says, calculate the mean growth rate of these larvae from 25 to 120 hours, and then include the units with your answer. So what we need to do is, remember, they have told us 25 and 120. At 120, I have about this point here, I have about 13.9, which is going to be millimeters. And at 25, I have, you can see, about 2.46 millimeters. So if I want to find the difference in length, it's going to be 13.9 minus 2.4, which gives me 11.44. And then down here, time of the time is going to be 120 hours minus 25 hours, which gives us 95 hours. And the growth rate is going to be, of course, the growth that has occurred divided by the time in which the growth has occurred. So 11.44 divided by 95 gives us 0.12 millimeters per hour as the growth rate. So this is the answer to that question. I will move on to the next part. The next part says, comment on the suitability of using this data to estimate the time of death. Here what we have to know is, remember, the method used is getting eggs and hatching them, and then observing the increase in size or the length of the larvae as time went by. So here I say, because the means experiments were repeated, the means were calculated, means experiments were repeated. However, we do not know the sample size, so since the sample size was not indicated, the results may not be that reliable. Another thing is about body temperature. Because they are incubating, they're incubating basically these the hatched larvae at a specific temperature that they suspected the body was found at. This could be misleading because the temperature change of the body could drop drastically, or it could drop slowly depending on the ambient temperature, or depending on whether the body was closed. That can be different, so it could lead to errors in the results. So I said, because body temperature after death may be different from the incubation temperature, since temperature changes after death, so that could be unreliable. And another part, for the first 45 hours, like I already said, the error bars were small, which is good. So this shows consistency in the results and reliability. However, after 45 hours, the error bars are too big, or they are big. So it is not suitable to use this method after 45 hours because there is not that much consistency due to the size of the deviation between the small and the greatest. Describe how the data shown in this graph could have been collected. So here they want us to basically say how the data would have been collected. So remember, they got the eggs and they hatched them, and then they incubated over a period of time. So what we have to do is we have to get several blowfly eggs. These have to be similar to those found on the dead body. And then we have to incubate in order to hatch. And then the length of these hatched blowfly larvae is going to be measured at intervals of a period of 120 hours. You're going to keep incubating for until 120 hours while periodically measuring the length of the larvae. So when we're doing this, the temperature has to be kept constant, the ambient temperature as well as the temperature within the experimentation. And then the nutrient supply has to be the same for all experiments. The species of the blowfly used have to be exactly the same for consistency in the results. And then we have to repeat the experiment at each specific at each specific condition, basically, the repeated experiment, and then we have to be able to measure the length, find the standard deviation, mean as well as the standard deviation, and then make a conclusion on the results to establish whether there is reliability or not. So the key thing is you have to use similar conditions for all experiments. You have to repeat every experiment, and then you have to calculate the mean length, and then you have to get the standard deviation in order to be able to plot showing the error bars and then make a conclusion on the reliability of the data. So lastly, from question 5 says, the time of death can be estimated using the body temperature of a corpse. A corpse is a dead body. So they say evaluate the use of the body temperature of a corpse to estimate the time of death. So if we are using the body temperature of a corpse, we have to make sure it is going to be measured. So it's going to be easy to use body temperature as a method of measurement. However, it has its own shortcomings. So the only advantage is the method is really easy to use or easy to measure, and there are a lot of calibration curves that are available online or by scientists that we can refer to. So we can use the calibration curves, and then we can work backwards in order to be able to successfully estimate the time of death. So using that, looking at how the body is losing temperature, you could find a suitable calibration curve and be able to work forward, predicting when the person basically died. However, there is a problem because body temperature depends on a lot of factors like ambient temperature. This is the temperature of the place where the body was found, and then of course whether the body was clothed or not. The conditions surrounding the body could lead to a rapid decrease or slow decrease in temperature, and this could greatly affect the results. So the method may not be that reliable because it has a lot of factors that are going to affect how the body loses temperature, and using temperature as the sole method for estimating time of death may not be a good idea. So this is the end of question 5. I will move on to question 6. Question 6, the diagram shows part of the carbon cycle. So for the carbon cycle, we can see here we have the atmosphere, we have the plants, so basically carbon back into the plants, from the plants back to the atmosphere, from the atmosphere to the surface of the ocean, and then from the surface of the ocean back to the atmosphere. We're going to look at the cycle. This is the carbon cycle. So they say the numbers in smooth brackets show the mass of carbon in petagrams are found in either the atmosphere, plants, or the surface ocean. The number in square brackets shows the mass of carbon in petagrams transferred between the atmosphere and the plants or surface ocean per year. This is per year. And then we go to this, of course, petagrams is 10 power 15 grams. Okay. The first question says, carbon is found in carbohydrates. The table shows some carbohydrates that may be found in plants and animals. The first question says, so the first part says, for each carbohydrate, put one cross in the appropriate box in each row to show where these carbohydrates are produced. So amylose, that is the first one. The carbohydrate is amylose. Amylose is produced in plants and not animals because, of course, amylose is part of starch. It's one of the molecules that make up starch. Of course, we know starch is made up of amylose and amylopectin. Since starch is made in plants, amylose should be made in plants as well. The next part is glucose. Glucose is going to be made in both plants and animals, as we can see. And then, because, of course, animals can break down glycogen into glucose, so basically we make glucose as well. And then glycogen, of course, is going to be only in animals. It's animals that store glycogen as a carbohydrate. Then starch is going to be made only in plants. So I will move on to the next part of this question. So B says the mass of carbon recycling between the atmosphere and the plants and the atmosphere and the oceans are roughly balanced. State how carbon is exchanged between the atmosphere and the ocean. Now the key thing is the carbon dioxide in the air will dissolve into the ocean, and then of course later on the carbon dioxide from the ocean will be released to the atmosphere. I would expect, like usually, since gases are going to dissolve in water, but when the temperature of the water increases, then the gases are going to dissolve. So carbon dioxide is going to, there is a cycle basically. From the air, it dissolves into the water, and then from the water, it could be dissolved and get out and basically into the atmosphere, depending on the conditions. So the answer is just that. Part B says calculate the increase in the mass of carbon in plants as a result of carbon exchange between the plants and the atmosphere in 1 year. So the increase I took to 120 minus 59, of course, everything in petagrams. And again, here they're asking for the increase in the mass of carbon in plants as a result of carbon exchange. Let us go back here and see. You can see there is a 120, and then the next part is going to be 120, and we have a 59. So 120 times 10 power 15 minus 59 times 10 power 15. So I put the 10 power 15 outside, but of course it's multiplying both of them. The difference I got was 61 times 10 power 15, and then it's going to be 6.1 times 10 power 16 when you move into get basically that to 1 decimal place. So writing it in standard form, it gives me that. However, remember, they wanted us to find the value in kilograms. And since this is 10 power 16 grams, to convert that to kilograms, you have to divide by 1,000. So divide by 1,000 gives us 6.1 times 10 power 13 kilograms. That is going to be the answer. So the next part says, describe how the carbon present in sugars in the plant is returned to the atmosphere in the carbon cycle. So if it's in the plants, returning to the atmosphere could be maybe plants could carry out respiration. As you guys know, plants do break down glucose in respiration, providing them with energy. And as a result, carbon dioxide and water is going to be produced. So that released carbon dioxide could go back to the atmosphere. Or animals could eat the carbon source found in the plants, and when we consume them, respiration occurs inside our bodies, producing basically, of course, carbon dioxide and water. So the first part I said, plants carry out respiration in their cells, releasing energy, and therefore glucose is used as a source of energy. Also, animals use glucose as a source of energy, and some microorganisms respire because they also use glucose as a source of energy. All these three processes, meaning plants, animals, and microorganisms, all their respiration is actually, if it's going to be aerobic, they all lead to production of carbon dioxide gas, which is released into the atmosphere in the process of basically respiration. Let's go to the next part. C. Anthropogenic climate change is a result of an increase in the mass of carbon in the atmosphere. State what is meant by the term anthropogenic climate change. So the anthropogenic part is to do with humans. And so basically this is like climate change as a result of human activity or human intervention. So I said it means change in temperature as a result of human activity. But this is climate change. I would say change in climate or change in the mean temperature. Mean temperature, this is it. As a result of human activity, that is the major part you're looking for. So lastly, the end of part 6, question 6 says, explain one way in which the effect of anthropogenic climate change can be reduced. How can we reduce climate change caused by humans? It means we have to decrease the amount of fossil fuels we use so that the amount of carbon dioxide released into the atmosphere decreases. As you know, methane is one good contributor towards, of course, climate change or towards global warming. So in this case, if we can decrease the cattle farming, reduce the amount of methane released into the atmosphere, that can be one good way. And lastly, we could practice reforestation. Instead of cutting down trees, at least if you cut down trees in an area, try to plant trees in order to have these carbon sinks, because as you know, trees do absorb this carbon dioxide. If we have so many trees that are planted, the carbon dioxide we release could be absorbed up by the trees, and that can be a good way of decreasing the concentration of CO2 in the atmosphere. So since this was just a 2-mark point, any of these would be okay for you to earn the marks you're looking for. This brings us to the end of question 6. I'll go straight to question 7. So question 7 says, stages of succession can be seen on different parts of a sand dune system. The greater the distance of the sand dune from the sea, the later the stage in succession. So, okay, they say the diagram shows a sand dune system, and the table gives some information about the different types of sand dunes in this system. So we have where the sea is, and then we have the forward dune, the embryo dune, the yellow dune, and the gray dune. And again, to go back, they say the greater the distance of the sand dune from the sea, the later the stage in succession. So the later the stage in succession distance from the sea, as distance increases from the sea. So the table here shows information about the soil depth, the percentage of organic matter, the pH, the percentage of bare ground, the number of different species, plant species, as well as the typical plant species. So here we're going to answer questions in relation to this. But as you can see, let me take you back up there. You can see, I want to concentrate on the gradient. You can see the gradient has greater amounts of soil in it. The percentage organic matter is a lot in comparison to the others, meaning there is a lot of life. The pH has greatly, of course, it's a little bit decreasing. The percentage bare ground, we see it's 10, meaning most of it is covered by plants. It's not that bare. The number of plant species, there are many plant species, and we can see the typical plant species. They range from different middle plants, meaning there is greater life, or there is more organisms in the gray dune in comparison to the forward dune. And again, I want to show you about the forward dune. When we see the forward dune here, you can see soil depth is really low, meaning it's like the colonization area where the beginners. Percentage organic matter is really low. The pH is kind of high. Percentage of bare ground, 97% bare. The number of species, there are just 2. And the typical plant species, it's sea rocket saltwater, meaning there is less occupancy, less organisms surviving within this area. So the gradient, there is more life thriving on there. So I will go straight to the first question. The pH of a solution is a measure of the concentration of hydrogen ions in that solution. So it is a log scale. Example, a solution of pH 5 contains 10 power negative 5 mole per decimeter cubed of hydrogen ions. What is the difference in concentration of hydrogen ions in a forward dune compared to the gray dune? If you look at the table, the forward dune had a pH equal to 8.5. And then the gray dune had a pH equal to 6.5. So if we want to convert pH to concentration of hydrogen ions, since pH is equal to the negative log of hydrogen ion concentration, if you take away the negative, so log of hydrogen ion concentration is equal to pH, negative of pH. And then this is to convert it to hydrogen ion concentration is going to be 10 power negative pH. So anytime you're given pH and you want to convert it to the concentration of hydrogen ions, you can use this thing I've circled here. So it means that the forward dune has a concentration of hydrogen ions of 10 power negative 8.5, which is that when you convert it to the concentration. And then this one here, the gray dune has a pH of a hydrogen ion concentration of 10 power negative 6.5. When you use your calculator, you get that. If you look at this value and this value, we can see it's 100 times that, meaning the answer for this part should be D. The forward dune has 100 times fewer hydrogen ions than the gray dune. So that is the answer. So the next part says, calculate the percentage increase in the number. Again, let me first remove this to create some space. Calculate the percentage increase in the number of plant species on the gray dune compared with the forward dune. Again, if we compare from the table, we can see that the forward dune has 2 species, while the gray dune has 15 species. So the increase in the number of species is going to be 15 minus 2, which gives us 13 species. Now, if we want to find the percentage increase, it will be the increase, which is 13, divided by the original, which is 2 times 100, giving us 650% as the percentage we're looking for. Let's go to the next part. The next part says, explain the changes in these sand dunes with distance from the sea. So remember, all the changes are shown in the table. So I took one change at a time. As distance from the sea increases, soil depth increases. We saw this in the table. Soil depth increases because they are older. Again, the dunes farthest away from the sea are the oldest dunes, and these dunes will have more humus. The increase in humus being deposited increases the depth of the soil. The other thing was, as distance increases from the sea, the percentage organic matter increases. As we know, organic matter increases because organic compounds have been decomposed. They die, and then they're decomposed by microorganisms, and as a result of that, they will be deposited onto the soil, increasing the organic matter. So here we said they had a lot of decomposition occurring by microorganisms, so that's it. The other thing is, as the distance from the sea increases, pH decreases. pH decreases because alkalinity decreases. The reason for that is there is more rain occurring and there is more humus being added. These two will dilute the soil, decreasing the mineral content basically, and leading to a decrease in alkalinity. The next part is as distance increases from the ocean or from the sea, the percentage of bare ground decreases. Bare ground decreases because there is more space being occupied by living organisms, meaning there is growth all over. The biggest percentage of the dune is actually enclosed by living organisms. That means growth occurs because there is a lot of humus, the soil is less less alkaline, and the decomposition of the dead pioneer species also creates more humus that covers the bare rock, increasing the possibility of the area to be occupied basically. And then as the distance increases from the sea, the number of different plant species increases. This is true. As more organic matter is deposited, as more humus is formed, and as the pH decreases down to an appropriate pH, the soil is more fertile. Of course, this is going to encourage growth of various plant species within this area. And lastly, as distance from the sea increases, the species of plants change. This is because changes in the soil conditions. So different species, those that will find this soil favorable are going to grow in this area. The different species that find the conditions available in these older dunes conducive will thrive, while the pioneer species that may find the conditions no longer favorable, they will be, of course, destroyed or they will die, and this will allow for succession to occur as many organisms occupy the sand dune. So let's continue to the next question. Sand dunes can be classified as fixed dunes, semi-fixed dunes, and shifting dunes. The graph shows the mean net productivity, primary productivity, NPP, above the ground and below the ground for these three types of sand dune. So here we can see there is of course, they've given us the key above the ground and below the ground, and we can see the data here. So we have the fixed dune, we can see the semi-fixed as well as the shifting dune. The first question says, describe the conclusion that can be drawn from this graph. Of course, from this graph, we can see the fixed dune, we can see above the ground, there is more mean NPP above the ground in the fixed dune, as well as in the semi-fixed dune, as well as in the shifting. So basically, above the ground, overall, there is greater NPP for all types of dunes. So, and then we can see, of course, there is lesser NPP for all types of for all types below the ground. That is the biggest thing we can see. So I say NPP above the ground and below the ground depend on the type of sand dune. Of course, we can see the sand dune has higher for all. So the NPP above the ground is always higher than the NPP below the ground. That is observable from the graph, as you can see. So I said overall, sand dunes have the highest NPP and shifting sand dunes have the least NPP. Or you'll say that basically the fixed sand dunes are going to have a higher than the shifting, and then or the shifting will have the least among the three overall. And then the shifting sand dunes has the largest percentage difference. Again, it's allowed for you to do some mathematics. Usually, you better do some mathematics. So in this case, actually, the third mark could be awarded for you doing some mathematics to show which one has a greater percentage difference. I did some calculations around here. You can see this part here, and I calculated the differences between all the three, and you can see 220 minus, of course, 70 gives us 150. 150 divided by 220 gives me 68.18. And then for this one here, 125 and 45, I got 64%, and then the 10 and 2.5 gave me 75%, meaning it has a percentage difference of 75% between NPP above the ground and that below the ground. So that can give you your third mark, or the mark. It can increase the marks, basically the marks you're trying to get. If you do some mathematics in any data you are given, usually calculated values are given marks. So let's continue. Lastly, they say explain how the products of the light-independent reactions become NPP below the ground. So the light-independent reactions, of course, these are the reactions in the Calvin cycle. How do these become NPP below the ground? You remember during the light-independent reactions, there is gulp, and gulp and glucose are going to be produced, As you know, glucose can be used to make sucrose, and some of the glucose can be used to make amino acids. Of course, when nitrates are combined, when both sucrose and amino acids. The reason as to why you're talking about sucrose and amino acids is you want to connect sucrose to the carbohydrate that is going to be stored, and the amino acids to the proteins and other products that are going to be stored, of course, underground. So I said both sucrose and amino acids can be transported to the roots through the phloem. And then lastly, these transported products are used to synthesize organic molecules that make up the biomass for NPP. Remember, amino acids are going to be basically for growth and increase in size, while the carbohydrates are going to be used as a storage, like starch to be stored as NPP below the ground. So basically these two things have to be talked about in this to be able to get these marks. So you have to include the whole process like I included it in here to be able to get all the marks you're looking for. Talk about how GALP is produced, how glucose is produced, how glucose is going to be used to make amino acids, or some glucose is going to be used to make sucrose, and how these are transported to the below the ground. And again, remember, this is below the ground, so you have to show they are going to be transported through the phloem, and then they can be used to synthesize. The amino acids are used to make proteins, and then the sucrose or the glucose can be used to make the carbohydrate, which could be starch, which is stored below the ground. So this brings us to the end of question 7. Let's go to the last question of this paper. Question 8. Patients with cystic fibrosis may develop serious bacterial infection. A patient with cystic fibrosis developed bacterial infection including Mycobacterium. A1. This patient was given a combination of antibiotics for several months. Explain why a combination of antibiotics had to be given for several months. So you guys, they give a combination because, of course, that some bacteria have resistance against antibiotics. So to be able to overcome that and to take into account that various bacteria could have infected this person, they give them a combination of antibiotics. I said a combination of antibiotics was given because different bacteria have resistance to different antibiotics. So giving several antibiotics ensures that there is enough dosage so basically given to the bacteria in order to ensure that they are all killed. This is, of course, to protect the person who is sick. Otherwise, this bacterial infection can do a lot of damage if the antibiotic cannot work on it. So part Roman 2 says this patient did not respond to the combination of antibiotics and later needed a lung transplant. Suggest why this patient needed a lung transplant. So in this case, and again, remember, Mycobacterium is really, really dangerous. This causes tuberculosis. So I say since Mycobacterium infects the lungs, failure to respond to a combination of antibiotics must have caused lung infection in the patient. So this led to the destruction of lung tissue. And again, you guys know when lung tissue is destroyed, the surface area for gaseous exchange is going to be greatly reduced. And if it's so, it means there is no sufficient oxygen supply to this person. So it's better for them to get a lung transplant because then they will have greater surface area and more oxygen can be passed into their bodies and carbon dioxide can be removed efficiently. So I say this greatly reduces the gaseous exchange surfaces, leading to insufficient oxygen supply to the blood. So a lung transplant would therefore ensure great surface area. So great surface area is present for sufficient gaseous exchange. So this is why the person actually got a lung transplant. They couldn't have enough surface area for oxygen, for basically absorbing in oxygen and releasing out carbon dioxide for their body to run smoothly. So a lung transplant is going to save them a lot of basically the stress they went through. You cannot breathe properly because you do not have enough oxygen, and that you have this infection. So it's kind of helping them to be able to supply their bodies with enough oxygen in order to live a healthier life. So let's go to the next part. So here we see, following the transplant, the patient was given immunosuppressant drugs. I hope you guys know this. The reason for this is when somebody gets an organ, the immune system is going to be able to fight. It's going to fight that organ because, of course, the organ is going to be foreign. So to protect the organ from being rejected, that patient or the person who received an organ is going to receive immunosuppressant drugs in order to prevent their immune system from rejecting the organ they have been given, basically. So they say immunosuppressant drugs weaken the immune system. Some of these drugs work by preventing DNA synthesis in the patient. As a result of the immunosuppressant drug treatment, the infection with Mycobacterium developed faster. Okay, so they say explain why the infection with Mycobacterium developed faster when the patient was taking immunosuppressant drugs. This is self-explanatory. The immunosuppressant drugs suppress your immune system, so the immune system is not going to be as active as possible, yet this person has some mycobacteria inside. Now, the immune system involves the T cells, the B cells, of course, the T killer cells, which are very important basically targeting these mycobacterium in order to destroy them. So it's going to be very problematic for the patient. So I said, taking immunosuppressant drugs prevents DNA synthesis. Now, this is problematic. If there is no DNA synthesis, this will inhibit mitosis. And if mitosis, because of course before mitosis occurs, we need to make copies of DNA. So if there is no DNA synthesis, the cell will not divide because there will not be enough genetic material for it to divide. So if mitosis is inhibited, there will be no clonal expansion of the T cells. And again, remember when you learned about the immune system, the T cells need to rapidly the T cells are going to cause the B cells to rapidly multiply to be able to produce the other cells or copies of themselves that can be able to produce the antibodies. And if the antibodies are not produced, it means there is no serious targeting or opsonization as well as destruction of some of these pathogens that have entered the body, in this case mycobacterium. So the clonal expansion we're talking about of T cells as well as B cells is to rapidly increase their number. We call it actually proliferation. They increase in number in order to kill the cells or the pathogens. So if they cannot increase, if there is no ability for mitosis to occur, these cells cannot increase in number. And it means since they're not increasing in number, there is no antibodies that can be rapidly produced. And if there are no antibodies, there is no opsonization. There is no destruction of the pathogen, or it's going to be actually delayed, or it's going to be very low. It means we have to depend only on the macrophages to destroy the mycobacteria, and it's actually going to be hard. So I said, so the macrophage cannot destroy the bacteria, hence infection is going to grow faster. Targeted destruction of the pathogen is going to be put down, and it means maybe we have to just depend on phagocytosis, which is going to be not enough to destroy some of these microorganisms. So let's continue on to the next. B. Phage therapy was used to treat the mycobacterium infection in this patient. Phages are viruses that target bacterial cells. The graph shows how the number of mycobacterium and the number of phages change following the start of phage therapy. So we can see these ones here are the bacteria, and then the dotted ones are the phages. Like you remember, they've said phages are going to be able to target the bacteria itself. So initially we see the number of bacteria is going to be really high, but as the phages begin to increase, the therapy has started, the numbers of the phages increase, we see the number of the bacteria begin to decrease. And then there is going to be this relationship whereby as one is decreasing, it's because they decrease and then increase and then decrease and then increase, basically continuously until they will be completely down. But let's go to the questions and see what they're asking us. So they say, explain the changes that in the number of bacteria and phages following the start of phage therapy. Use the information in the graph to support your answer. So this is a 6-mark question. You have to write out as much as you can and look at the data and how everything goes in order to be able to obtain it. One thing we have to know is when you look at the two graphs, they are showing a cyclic pattern, and that cyclic pattern is showing you that as one increases, the other is going to, the increasing one is going to cause one to decrease. So as the phages increase, they cause the bacteria to decrease because they are infecting them basically. So I said at the start of phage therapy, there was still an increase in the number of bacteria. The reason for this is because the phages were not yet in their lytic phase. I think you know the lytic cycle. Phages are going to increase. They're going to go infect a cell, stay in there basically until until they are triggered. Basically, when they are triggered, they are going to rapidly produce copies of themselves, and then the lytic cycle will occur whereby they break open the bacterial cell and they are released in numbers. Then they begin to attack newer cells. So here I say there was still an increase in the number of bacteria because the lytic cycle takes time. So the lytic cycle of these phages is going to take time. It takes longer to be able to reach that phase. So I say the virus numbers begin to increase, and as the bacteria decrease in number, this is because the viruses infect the bacteria, replicating inside them and cause them to burst. This is when they have reached that lytic phase, when they burst out. This bursting will destroy the bacteria, and the number of the viruses will increase. The number of viruses increases because they burst out of the bacteria in thousands and thousands. So the numbers are going to be greatly high. So the number of viruses increase, that of the bacteria, then that of the bacteria basically will decrease. And then the virus begins to decrease the number because they get engulfed by macrophages. Of course, you know, the macrophage is going to engulf them. They will be destroyed, and there will be another repeating cycle. Some of them, as most of them are destroyed, some of them are going to be able to infect newer bacterial cells. And over time, those bacterial cells will also reach another lytic cycle where they come out, and then the macrophages will destroy them. So it's going to be a repeating cycle. So I say this repeating cyclic increase and decrease in the number of bacteria, since not all bacteria get infected, of course, and when the bacteria increase, the virus increase and decrease in a cyclic pattern because not all of them, of course, are destroyed. And again, not all bacteria are going to be destroyed, and not all viruses are going to be destroyed by the macrophages. So it's this cycle, because when some of the viruses burst open out of the cells, it doesn't mean that every cell has gone through that lytic cycle. They may not go through the lytic cycle at the same time, and it means there are going to be some remaining viruses in there and some remaining bacteria. So it's going to be a continuous cycle. So I say the viruses increase and decrease in cyclic pattern because not all of them can be destroyed by the macrophages, and they continue to infect new cells, and eventually there are no viruses and no bacteria. I try to put this graph here so that you can see. You can see in the end it reaches almost zero level, meaning they are completely destroyed. So that decrease is going to go like that, like that, like that, like that, until they are completely destroyed. At that time, there is no more infection, basically. So this brings us to the end of this paper. Thank you for being with us, and we hope to see you in our next video. Please do remember to subscribe, and please do comment. See you next time. Bye-bye.